All right, here in our second video clip, I'm going to talk a little bit about, and you guys can read the summaries here on the slides, but I'm going to talk about a few example viruses just so you can see some of the diversity among viruses in terms of how they achieve all these different steps of their life cycles. And the first example we're going to talk about are the herpes viruses. So of course we've all heard of herpes viruses before. The herpes viruses are a pretty big family of viruses, and of course they do include the viruses that cause genital herpes, but also oral herpes. If you've ever had a fever blister before, those are caused by types of herpes viruses. The chickenpox virus is a member of this family. They're all closely related to each other. And also Epstein-Barr virus, which causes mononucleosis, that's also a member of this particular family as well. All right, so these types of viruses all tend to kind of replicate themselves in a very similar way, but they go to different locations in the body, and it's often due to that host cell receptor. They're different members of this herpes virus family will attach to different cell types in the body because they use different receptors to attach to. All right, so what we're seeing on this diagram over here, the viruses are, of course, have to attach to the host cell, and then they do this membrane fusion. That's where these are enveloped viruses. So the envelope melts or fuses with the host cell's membrane, and then the capsid on the inside is released inside the host cell. This is a DNA virus, double-stranded DNA virus, so the DNA is inside that capsid. And then what they're showing you here is the capsid is actually going to go inside the nucleus of the host cell, and the capsids actually slide. Remember how you maybe learned about the cytoskeleton that exists inside our cells before? And it will slide along protein fibers of the cytoskeleton and into the nucleus. It'll pass through a nuclear pore and go inside there. Then once inside there, the viral DNA comes out. The DNA genome comes out of the capsid. And then really what happens then, the DNA is in the nucleus of the host cell, and the host cell is more or less kind of treating it like it's its own DNA. So you have transcription that takes place there. You're making messenger RNAs. Don't worry so much about early and late. All that means is there are some messenger RNAs made early during the virus's replication cycle and some that are made later. Those messenger RNAs go out here to the cytoplasm, and you do translation. You build the proteins. That's why it says protein synthesis there. All right, and then you notice we've got an arrow here because a lot of those viral proteins have to go back inside the nucleus so that you can package up and make new capsids. So the DNA is going to be copied in the nucleus, and these capsid proteins have to also go in the nucleus and assemble new capsids. And then over here what you see is they're showing you the endoplasmic reticulum. That's a membrane covered compartment, an organelle inside eukaryotic type cells. And in the past you may have learned that, depending on what classes you've had, that the endoplasmic reticulum is a site where proteins that are going to be secreted are first produced. Also proteins that are going to wind up on the cell surface are produced there as well. They kind of go through an assembly line process where they go from the endoplasmic reticulum to the Golgi and up to the cell surface in order to get out. So what the herpes viruses do, they've hijacked that natural process. And so we talked about budding on the last video clip. Herpes viruses are enveloped, so they have to bud to pick up their envelope. The herpes viruses bud into the ER, these little membrane covered compartments inside the host cell, and that's how they pick up their envelope. They don't actually bud at the cell surface. And then from there they go through the assembly line up here to the cell surface, and they're secreted. Just like when you learn about in biology 202, endocrine cells that make hormones, many of those are proteins that get secreted to the outside of the cell. So more or less, herpes viruses trick the host cell into thinking that it's secreting something that it naturally makes, but instead it's secreting these herpes viruses, and that's how they wind up getting out of their host cell and then floating off to find something else to infect, another host cell to infect. So they're kind of sneaky. All right, another viral example I want to talk about are the influenza viruses. And the influenza viruses are in the negative strand RNA virus category. And another interesting thing about flu viruses, influenza, is they have segmented RNA genomes. And what that means is their genome is not just in one piece. It's divided up into 8 pieces. Kind of like the human genome is divided up into 23 chromosomes. And then our cells have 2 copies of those. So you have 46 in total. Influenza has a segmented genome. It actually has 8 pieces of RNA. And they are pieces of negative strand RNA. So remember, when that means when they go inside the host cell first has to, or they first have to be converted to plus strands, because the plus strand versions are the ones that host cells read as messenger RNA and can do translation with. Also, if you're going to build more of these minus strand genomes, you've got to go from the minuses to the plus strand copies to the new minus strand. You go from minus to plus to minus. The RNA polymerase enzymes that do that have to go through those steps to make the new genomes. Okay, I know a really interesting thing about influenza. You guys know that every year, the flu vaccine gets replaced with a new flu vaccine because the strains of influenza that are affecting the world from year to year vary quite a bit. And one of the main reasons for that, there are 2 reasons for that. One is, okay, so we talked about how DNA polymerase enzymes are good proofreaders. So if they make errors during replication, they tend to get proofread, they tend to get corrected. The RNA polymerases of RNA viruses are generally not good proofreaders, so they tend to mutate very quickly. Okay, so if these RNA genome pieces are mutating, those have genes on them that code for, here on influenza, you've got all these little protein spikes in the surface. Those are flu proteins. So if the genes are mutating, those flu protein spikes there mutate. That's what your immune system is looking at. Your immune system recognizes influenza by these protein spikes on the surface. So if flu is changing its spikes pretty easily, that means, all right, if you had the flu in 2012, you have immunity to flu from 2012, but meanwhile influenza has mutated over the past couple of years and has slightly altered the little spikes on its surface, and now your immune system can't recognize it anymore. Another thing about influenza, though, that's interesting is because it is divided, its genome is divided up into these 8 segments or pieces. It can go through some pretty big alterations in cases where a single cell in a host gets infected simultaneously with 2 different strains of the flu, because they can wind up swapping gene segments. That's what happened a few years ago when we had the H1N1 flu outbreak. You had, in Mexico, it was traced to Mexico, there apparently was a little pig down there, and the pig, one cell in the pig simultaneously was infected with a human flu virus strain, which had 8 little pieces of RNA, 4, 5, 6, 7, 8, and at the same time it was infected with another flu strain, which was a bird flu, swine flu combination. There are flu strains that replicate really well in humans, some replicate really well in birds, and some prefer pigs as their hosts, and you get these weird combinations of those different types. So inside in a pig, in a single cell, inside a pig in Mexico, these two types of flu viruses got together and started replicating at the same time. And as you assembled new viruses, you wind up with different combinations of these 8 pieces. Like you might have a virus that picked up a copy of that one, and that one, and that piece, and this piece, and that piece, and so forth and so on. You can imagine if all those pieces of RNA were replicating at the same time in the host cell, and each new virus particle only picks up 8 of them, you know, 1 copy of each segment, you're going to get different combinations that come out. And unfortunately, every once in a while, when that type of genetic shuffling takes place, a new strain of flu emerges that's more severe, and almost nobody has immunity to it. And so that's where this H1N1 flu came from a few years ago. Then a farmer in Mexico picked it up, and then he started spreading it to other people, and it spread across the whole world from there. Fortunately, that H1N1 flu was not as severe as was originally feared when it first emerged, but every once in a while there are new strains of the flu that emerge that are very severe, and almost nobody has immunity to it. And there's a lot of fear that sometime down the road we're going to have a really bad worldwide outbreak of influenza with some really severe strain of flu that will be very difficult to treat. All right, then a third type of virus to talk about, and I've already mentioned a little bit about this, human immunodeficiency virus, or HIV. It's an example of a retrovirus. And again, that means that inside the virus you have an RNA genome, but once it goes inside the host cell, it gets converted to double-stranded DNA. And as I mentioned before, that is the reverse of transcription backwards. So HIV has an enzyme called reverse transcriptase that performs that process. Our cells don't have those enzymes. So the reverse transcriptase enzymes of HIV are a very good target for drugs. And in fact, many of the drugs that are used to treat patients who are infected with HIV do interfere with reverse transcriptase. So we've got some good drugs that will screw up reverse transcriptase, and they don't screw up any host cell enzymes or host cell processes. So they're pretty good drugs to take at knocking down HIV. Unfortunately, they don't completely wipe it out, but they're very good for keeping it under control. Okay, so on our diagram over here, HIV's life cycle or replication cycle over here. HIV first attaches to a host cell receptor called CD4. We talked about that on Wednesday. There's also, on this diagram, they're showing you that there is a co-receptor. That's another molecule on the cell surface that HIV attaches to. And that is, there are 2 kinds that it can use. One's called CCR5, one's called CXCR4. That's why that's shown there on the diagram. So CD4 and that other co-receptor, HIV actually uses both of those to go inside the host cell. And you may have heard before that there are people who are kind of naturally resistant to HIV. HIV doesn't infect their cells very well, and it's because one of these co-receptors is mutated in those people. So HIV can't enter the host cells in those people as easily. All right, once HIV attaches, it undergoes this membrane fusion process, and there's its capsid, this thing that kind of looks like a thimble, comes out. Reverse transcriptase does its job and converts the RNA genome to double-stranded DNA, like you see there. And then HIV goes through a process called integration. That's what they're showing you right here. That double-stranded version of HIV's genome gets integrated, it gets inserted into the host cell's DNA, into your own chromosomes somewhere, which is kind of sneaky. And HIV actually has an enzyme that cuts your DNA, hopefully not your personal DNA, but it will cut the host cell's DNA and actually insert HIV into your DNA. Then the host cell just thinks that HIV's genome is one of its own genes, and so it starts or a series of genes, because HIV has more than one gene. So those genes get transcribed. You make messenger RNAs. Those go out into the cytoplasm, and you have translation that takes place. So you make the new proteins of HIV. So you package up new HIV genomes that are RNA with all those HIV proteins to make the capsid. And then it pushes itself through the cell membrane. It does budding, and it picks up an envelope, and it's also going to pick up some more of those GP120 spikes that we talked about earlier. So, and again, this reverse transcriptase is a pretty good target for anti-HIV drug therapy, because our cells don't rely on reverse transcriptase enzymes. Actually, the integrase enzyme, the one that takes the HIV DNA and puts it into the host cell's DNA, that's another good target. And there are some other enzymes of HIV that we have pretty good drugs that will screw those enzymes up as well. Lots of research has gone into coming up with drugs to treat HIV. So HIV is kind of unique. It's a virus where we actually do have several different kinds of drugs to treat people who are infected with it. But again, the problem is these drugs do a good job of beating HIV down. HIV, though, can go dormant because it actually integrates its DNA genome into a host cell's DNA. It's very good at going dormant, and the dormant virus-infected cells really can't be killed by the drugs. We don't have a way of finding those dormant viral genomes yet, unfortunately. And until we do, it's going to be very difficult to actually cure somebody of an HIV infection. Okay, so when you are reviewing virus replication, just keep in mind all viruses have certain things in common that they have to do. They've all got to enter a host cell. They're all going to use the host cell's machinery to replicate themselves, and especially the ribosomes. Viruses do not have their own ribosomes. That's a key reason they need a host cell, because you got to have ribosomes to make proteins. So they don't have their own ribosomes. They don't have their own tRNAs. They don't have their own amino acids floating around that they can use to build their proteins. So they're going to hijack those things from their host cell. Then the third thing that they have in common is exiting. All viruses have to exit a host, and they will do that either by budding or lysis. Enveloped viruses bud. Non-enveloped viruses are generally going to get out by causing the host cell to burst. Some ways that viruses differ from one another: they have different host cell receptors. Their genomes, DNA or RNA, and then even, if you're an RNA virus, are you a plus strand virus? Are you a minus strand virus? If you're a DNA virus, is it double-stranded DNA? Is it single-stranded DNA? Lots of variation. Viruses don't follow the central dogma always that we learned about of going from DNA to RNA to protein. As you've seen, there are RNA viruses that go from RNA to RNA to protein, and they never even have DNA as part of their life cycle. So that's very unique. So just go back and review the different possible variations and review those example virus life cycles that I just went over as you're going over this particular topic. I will stop this particular video clip there, and then I'm going to do one more on viruses to finish out Chapter 5.